I'm Harry Glurkian, and this is Moneyball Medicine, the interview podcast where we meet researchers, entrepreneurs, and physicians who are using the power of data to improve patient health and make healthcare delivery more efficient. You can think of each episode as a new chapter in the never-ending audio version of my 2017 book, Moneyball Medicine, Thriving in the New Data-Driven Healthcare Market. Well, hello, everybody, and welcome to today's show.
Thank you.
Thank you. It's great to be here.
Yeah, it's great to have all of you here. For everybody listening and watching, we were actually supposed to do this in person, but unfortunately, the Delta variant sort of threw a monkey wrench in that whole process. So I reserve the right that we can do this in the future and actually get together when this whole thing is over, like normal human beings. Look, each of you are working on super exciting things, different companies focusing in different areas. So if, and I know you all know each other, so I'm going to step back 1 second and say, if you had to give a brief description of your company or, pretend you don't know each other, we're at a cocktail party. And you're going to give me 2 or 3 sentences about what you're doing and why it's interesting, how would you sort of do that? And Andrew, since you're the ringleader that sort of helped bring this group together, I'll throw it out to you first to sort of get going.
Well, that's a lot of pressure, but so certainly, like our description, I think is pretty simple. We are a preclinical stage pharmaceutical company. And we happen to have a proprietary artificial intelligence platform that's discovered all the assets that we have under development. And these days we have 18 programs, 18 different disease areas where we've got new experimental medications, and we are working on progressing those new inventions to the clinic and ultimately to FDA approval.
Peyton?
Hi, everyone. I'm Peyton and one of the co-founders of BigHat Biosciences. And our mission is to improve human health by making it easier to design advanced antibody therapeutics. So we actually do that through a combination of a high-speed wet lab and machine learning techniques in order to very iteratively design and improve antibodies until they meet unmet patient need. And it's been a lot of fun, and we've been founded since 2019.
And finally, Tim.
Thanks for the opportunity, Harry. So Inflammatix was founded about 5 years ago, spun out of Stanford, along with, of course, Aria and BigHat. We are designing novel diagnostics focused on acute care and critical illness needs. So we basically have a data analytics platform that allows us to decode certain signals of gene expression within the immune system. And then for those of you watching, I'll show you, we have a cartridge that allows us to sort of implement that in a 30-minute point of care diagnostic setting. So our particular focus is basically bringing precision medicine into acute care settings, the hospital, the clinic, the ICU, where sort of historically there hasn't been a lot of diagnostic innovation.
Interesting. It's funny because I wrote a textbook on how to commercialize novel diagnostics a few years ago, because unless you've been through the ringer, you may not know all the different pieces. But you guys now all know each other. Now that may not surprising because we're in Silicon Valley and I'm actually in Berkeley right now, but that's close enough. And drug discovery companies and tech companies are all swimming around each other, but your connection is a little bit deeper. I mean, you guys all went to Stanford together. So this is not necessarily a commercial for Stanford, but that's pretty interesting that 3 CEOs of data-driven, healthcare companies out of the same class, whoosh, come out of Stanford. So how did you guys meet at first?
Well, and I would say we're not the only ones. It's just, the people that happen to be in front of you today. It's funny. So right before this, I sent a panicked email because I didn't want to say something that wasn't true. I was like, Peyton, you were in this class, weren't you? To which the answer is yes.
If I was Andrew's TA, or because we'd all actually met in the same class. So I think our Stanford journeys all started, it sounds like the same year.
About the same time. Yeah, I think the same year.
Same time. And we all were taking translational bioinformatics, which was a course taught by, I believe, Atul Butte, who I think, really brought to fame the idea of big data for biology, what you can draw out of very large data sets and drawing insights. And so we were all in the same class and with many other people, as Andrew said, and it was a lot of fun. I think it was the start of, now I guess long journeys for all of us in a similar vein.
Yeah, and it was a place where just like, I think what was awesome about that class, again, not to be an advertisement for the coursework, but it was kind of my characterization of the class was you basically learned how other people use data science to solve some medical mystery, like across the spectrum. And so the purpose of learning all of that was to just kind of fill you full of ideas of things that you could do. And then the capstone of the class was, a final project where you basically had to come up with something, right? And so you were just sort of primed with all this like super interesting sort of research on how other people had approached like very different problems in the space. And for me, it was just the source of lots of interesting ideas that then, helped me ultimately create what's the technology behind our company today.
It is remarkable how much came out of Stanford Biomedical Informatics, though. I mean, to Andrew's point, there are a number of other CEOs that came through in that sort of in a several, maybe 5 or 7 year stretch, all out of the same program. And I think a lot of it had to do with that, yes, this one particular class had all the different applications of data science sort of across the spectrum of life sciences, but they also attracted people like that, right? I mean, everyone on this call has a very different background before Stanford BMI, and I think that was part of what made that culture so special, is that it ends up being a real team sport, whether your background was medicine or business or math or computer science or bioengineering or anything else, and sort of that, learning a technique from A and applying it into area B, I think was a pretty successful way to grow innovation.
I feel like a venture guy, I should be standing at the exit door and just sort of saying, what's your idea? What's your idea? And screening as if.
Funny enough, some folks have also become venture capitalists.
Yep.
So you can add that to the list. Well,
That's true. Yep.
Yeah.
Yep.
So, was there anything in particular that you guys like, interests or questions or discussions that you sort of bonded over that sort of brought you together? I mean, even as just friends that decided to keep in touch?
Well, I think it's probably different for different people. I think the first real interaction I had with Tim, the details escape me because this is almost 10 years ago now, but I remember, like, he's a medical doctor, right? He's got a MD and a PhD, if I'm not mistaken. And so I'm a hardcore computer scientist. That's my background. And so back in those days, I was rapidly learning all I could about medicine and biology. And I remember the topic, but I do recall him, helping me after class with something that wasn't just quite, sitting in my head correctly. And I remember thinking like, what a nice dude to kind of take some time and give me a little private tutoring. And then if I recall afterwards, he said, yeah, so I'm trying to do this stuff with some clinical data. Can you help me, with this sort of stuff, which if I remember correctly, I never actually helped you. I remember talking about how I might be able to help you. And then eventually you said, I figured it out. I don't need you.
We were trying to build a web scraper. And I said, I have no idea how to.
Oh yeah, totally done that. Yeah, I've done that lots of times. So yeah, so like that's how the conversation started with Tim, was sort of, I think to the point about having very different backgrounds. And with Peyton, I don't really recall the first interaction. I remember we were in a journal club, maybe with Russ, and you were talking about some stuff. But I think the more I got connected to her was around the time she was working on her defense, and actually went to her PhD defense. And I have this this BS detector that sometimes goes off a little early, right? When people make a statement, I'm like, I don't know about that. And I remember sitting in her defense, and every time she said something that made me, do one of these, like, wait a minute kind of stuff, she instantly resolved it in the next sentence. I was like, okay, all right, that's cool.
Oh, that feels good.
Yeah.
And so, I passed, fortunately.
Well, you don't have to pass my scrutiny, obviously, but yeah, I think that led to a number of kind of interesting conversations as she was contemplating what to do next. As she was moving through her career, but yeah, I think that the interactions are very, very different for each person. At least that's my view, but I don't know if you guys have different memories. But
yeah, I think what's interesting, I mean, just generally, I agree with that. And I think one of the most interesting parts of BMI is, Tim said, is just the backgrounds that everyone has. And I have this, so, I also come from the kind of applied math, computer science background. And there's this kind of fascination of what you can do, right, with kind of computational skills in biology. I think to me, a lot of the conversations around like, where do I even apply this to? I think people sort of think of computational biology as a maybe sort of a niche small field at the intersection of maybe somewhere in where biology meets I guess statistics, computer science, and math. But it's so broad and it's so vast. And I think most of the I'd say most exciting conversations I've had are what are you in? We work in immunology. You're a clinician. You work with clinical data. How do you apply these tools? Like what the most both daunting but fun task upon showing up, I think, at Stanford was such an incredible ecosystem here is where do you even focus your attention? Like where should you work? There's too many exciting opportunities to pick. And I think some of the fun conversations I remember I'm also having with Tim with more clinical background. It's like what's actually useful? I want to do something useful and sort of trying to figure out where this where you can actually kind of apply your time to the most impactful problem. It was a lot of fun.
And I think, Tim, it'd be great for you to share. I mean, when we first met, I'd asked you kind of like, what were you doing there? What your story was? I can't remember the words, but you basically said like, look, I'm a surgeon, if I recall, and I'm trying to like save people's lives. And I'm just thinking like, is there a better way? Can I like just, do something that's going to have a much larger impact? And I don't know what that is yet. I know, wildly paraphrasing what you said, right? But like, I'm thinking about like what that could be. And I think that, when I met you, you were sort of on the hunt for figuring out where to apply, kind of the skill set.
I mean, I think that everyone shows up with their strengths and weaknesses. Mine certainly was. The summer before the program actually started, I had to take, sort of like basic courses in computer science and linear algebra. And I remember, I literally went from my last overnight call at Santa Clara Valley Medical Center, running 2 ICUs, to the next morning, CS 106X, which, because it was the summer, was filled with all the high school students that are just like total whiz kids. So I was saying with, like 16-year-olds, and they're like, we're learning like order of operations or something, and they're raising their hands, and I'm like desperately trying to write down like O of N means. And obviously Andrew and Peyton were among the folks that sort of helped me on the basic sciences side of things. But I think that the story about sort of getting the question right is absolutely correct. And I remember actually the time that I knew I was in the right program was maybe 2 or 3 months into training. I was at they used to have these like sort of work in progress talks, and it was like, Wednesday or Thursday or something, you bring your lunch. And somebody was talking about this thing that sounded very, very cool to me. It was all about how you could program a system to learn new knowledge on its own. And it was like, generalized AI for health data. And I was incredibly impressed. And the first example that was given was like, so we've sifted through all of the billions of data points, and I have discovered, and he stumbles over the drug name. And I've discovered that clopidogrel, which he meant clopidogrel, is associated with bleeding events. And everyone goes, and I was like, put my hand up, like, that's an antiplatelet medication. And he looks at me and I'm like, so the point of that is that it thins the blood. And he looks at me and he's like, so bleeding is a known side effect. Totally crestfallen. Like, people knew this already? Like, I had no idea. I was like, I do have something to contribute. So it's a good merge. Yeah.
Well, so, Tim, you're running a diagnostics company. Peyton and Andrew, you're running what I'll lump together as drug discovery companies. And different markets, different regulatory processes, but I'm sure there are common challenges to life science startups in the Valley. So, what are some of the biggest challenges that you guys see? Is it scalability? Is it finding the right people? Is it finding the right investors? Where do you guys see your challenges?
And I would just say, for a little clarification to Peyton's point about there's so many different problems, even though Peyton and I are both in the business of creating new medicines, we couldn't be any more different. We're a small molecule company. She's a large molecule company. If you know what that means,
yes.
I'm making motorcycles, she's making trucks. Like we're just doing completely different things. And so there's, I think some of the to your question about like kind of what are the very similar things, but like we're not really even competing with one another from that perspective. But I think to answer your question, at least from my viewpoint, you kind of have to do all those things. I think, startups, sort of everything has to work. You can't sort of have any one thing that doesn't function. And whether that's the science or the fundraising or the team or all of those things, if you've got a problem in any one of those areas, it can be life-threatening to the company. And so I think part of the experience for the entrepreneur is sort of, because your time is limited and your resources limited, is sort of finding a best fit to try to solve, or to maximize all of those problems simultaneously. And I would say all the things that you've listed have, they all at various points in the company, have been critical. And it's more of a juggling act rather than, geez, all you need to do is just knock it out of the park on, financing, and who cares about anything else? We know lots of stories where that, hasn't gone well, or you knock it on the park on an exceptional team and, like the other things don't come together. So, from my standpoint, all that stuff has to work, all of it.
I think my answer continues. I think one of the things and many people who just find, I would say, many scientific inquiries fascinating, is just what to work on. And I have the same problem now. I think it happened when I went to Stanford, and happened, postdoc, and happened now. And in the context of my company, so we basically have a platform that can work on engineering any protein. We work on antibodies, but really can be anything. And so, we have this basically landscape where there are tons of diseases with unmet need. There's sort of tons of opportunities for the type of therapeutic protein you would use, whether that's a standard antibody, monoclonal IgG, sort of a next generation antibody. And so we always have to decide, what are the programs going to be? What are you going to go after? What's the modality? And I think, that is at the crux of it, like, a drug discovery company, which is what is the shape of your company. But our platform is so broad that basically we can work on so many things. And I, once again, find myself facing the same problem, which is, okay, like, where should we focus our attention? And that's been really fun, I think, and getting more also into the, I guess maybe more of Tim's background, but sort of learning more about the clinical side of things and where unmet need is and where that pairs with our technology. But I agree with what Andrew said. Nothing really can go shortchanged, but that's been, the same theme, I would say, just now in a different vein.
Yeah, I think about this as a balance, right, of dynamics where you're at different stages at different points, depending on where you are in the development cycle, and you need different people, and different issues become a problem at different points, or maybe become more acute at different points. But all of you guys have one theme in common, which is why we're on the show together, is data and some form of machine learning or other part of artificial intelligence that's being applied to find something valuable, or identify some valuable piece of information that can make something actionable. How do you, each of you, it's sort of a big question, but how do you employ machine learning and AI in what you're doing in each of your businesses? Because I think of these things as like, I have a toolbox, and then I have to apply that tool in a very specific way with a specific set of knowledge that can feed it, where I can get an output that I'm looking for. And so each one of you, like you said, Andrew, you're working on the motorcycle, she's working on the big truck, and he's trying to make sure that everybody gets diagnosed and not ends up in worse than they already are. So how are you, each of you, thinking or approaching this in your own unique way, if you can summarize it? Tim, why don't you go first? Because
sure. Well, so for us, each one of our tests works by measuring a discrete number of genes within the body. It's their expression levels. So for instance, for our flagship test, INSEP, we look at 29 different gene expression levels from blood. And then of course, we have to somehow integrate that 29 different levels into actionable information. And so the back end of that is the data science part, the machine learning. So the step one is actually choosing what to measure. And then after you've chosen what to measure, then it's training hardened algorithms that turn 29 different things into a score that says, like, this person has a bacterial infection. And then of course, doing that repeatably, right? Doing it in a way that is traceable and verifiable, and then all of the post hoc, how is it affected by different demographics, and how is it, in the actual context of care. And of course, in the coming years, when actually implemented in a health system, how does it impact patients and providers, and does it save costs and improve outcomes? And maybe just to, since I didn't get a chance to answer, I think one of the questions about challenges is a lot of times sort of it changes with sort of the application that you're taking farther, right? One of the things that we all have in common, I think, is that we're all platform companies. And to Peyton's point, like, you can apply that data science platform to a lot of different areas, but each one of those areas has to be taken through a very long development process to actually help a person. And the challenges totally change along that development lifecycle.
And just for everybody listening, so you developed this product. What is the so what? What is the impact?
Yeah, I mean, in our case, we decided that we wanted to go after one first indication that would be a big enough hit to make the business matter. We've got lots of things we'd like to do in the long run, but sepsis is an area of outstanding unmet need. And the so what is right now, if you go in and you're feeling sick and you see a doctor and you want to know, hey doc, like, do I need antibiotics? There is literally no test that can answer that question. It's a guess. So it's not to say that antibiotics aren't administered quickly, but as a physician myself, I can tell you that is, it's a guess at first. And then you have to wait for tests to come back, and those tests themselves are imperfect. And so something like 40% of antibiotics are probably misprescribed. And if you knew in 30 minutes, hey, this person has a bacterial infection or not, you could greatly simplify care and really improve outcomes. And that's the promise. But the challenge, of course, is that beyond the data science, there's so much that goes into, building the product and proving out the clinical data and getting it through FDA and then getting it reimbursed and getting it rolled out more broadly. If you want to get to the point where you've actually helped a number of people and built a solid business.
Yeah, we did. In my last company before I moved on to venture, we had a strategy consulting firm, and we did a lot of digging into sepsis. That was a big problem that people were trying to crack. And if you could crack it, it's the opportunity is quite significant.
Yeah.
So yeah, Peyton, Andrew, how do you guys think about it? Because I'm thinking manipulating an antibody and sort of tweaking little parts of it until you find the exact fit. Supercomputing or massive computing.
It's funny. I actually think that the context in which we all met, which is, when I think big data was becoming really popular in medicine, is actually a great context, I think, for where BigHat ended up. And it's funny because it's been kind of a long journey. It always happens when I look back, you're like, yeah, that makes sense, right? Based on where I was. And so, we actually, put a lot of our attention into integrating the wet lab with the dry lab component. And this is actually, with a goal of making big data into what I might call sort of smart data or agile data, which is that, the idea back in the day when first, I would say you got tons and tons of really large data sets and you can sort of mine them, right? You can look for trends, you can sort of figure out something, interesting relationship between gene expression and patient outcome. And I kept throughout my career feeling frustrated by being handed the data set and sort of having to just mine and not having kind of, ownership of being able to say, I want to look here. I want more data here, right? You're sort of handed a really large data set and you're now kind of like a passenger in this kind of, a data set that's already been generated. You cannot modify it once it's been made. That's kind of this fixed data setting. And, as a computational person, that you're often the second person, like a wet lab or experimental lab is making this data and you kind of get it, right? And so, throughout, I would say, especially at my time at Stanford, this was very much the case where I felt kind of trapped in being given a data set that, I didn't necessarily design, but I could sort of mine. And so at BigHat, we're basically trying to now put computation in the driver's seat. And kind of change that paradigm where actually now, instead of just getting one large data set you design up front, you acknowledge that biology and science are very iterative, right? As you said, you sort of start with an antibody sequence, but would you stop there? If you could just make one tweak, maybe you'd make it 10x better, 100x better with 2 tweaks. So how do you enable that? How do you enable that very rapid cycling? And so we view this as kind of the intersection of how closely can the lab and the computational side interact, and how can they inform each other? How can one learn from the other? And so we actually, enable a computational person to design an antibody on Monday in a few days. You synthesize, purify, characterize the antibody, and kind of, understand, are you moving in the right direction or are you not? And repeat, and then repeat and repeat and repeat. So you don't get kind of stuck in this fixed data setting. And so this is really attractive for a lot of ways, right? There are a lot of reasons. You kind of can end up in a really good regime in this big data or sort of area, but there's kind of a lot of lost opportunity in terms of being able to kind of be very agile and move towards something that looks promising and iterate more. And the goal is that that will allow us to enable kind of different types of antibodies that don't even exist today, because you can't engineer them that easily, right? You kind of are stuck with a fixed format. So it's been really fun. And so we've been spending a lot of time designing the wet lab to kind of support the machine learning side and data science side from the ground up, and vice versa. And so it's a pretty unique sort of setup. And I like to think of it as sort of smart data, right? You're now thinking really closely about what should I generate that will be helpful, and can use that to inform how you redesign the next data set and improve your antibody every time, in our case.
Andrew?
Yeah, it's interesting to hear the different stories. I think all of us are kind of taking the approach that, what data sources and what artificial intelligence allows you to do is to take real world data and then make some prediction under uncertainty, with the expectation that prediction is potentially better than what you could do with other methods. And so, kind of tying this back to when I was a student and thinking about, where are the places I could make a big impact, was very interesting to me was very complex disease and disease where there was really no single biomedical measurement that would help kind of unravel the mystery of the biology behind that disease, and therefore could, explain something about pathogenesis that would lead to a new discovery or a new medication as a result. And part of that coursework in 2017 was this concept of integrative genomics, this idea of using different data sources that are all keyed to the same thing, maybe a gene or a gene product, and kind of looking for that overlapping evidence. And there were some great papers that were shown. There was one, I think, by Eric Lander in particular, where he was using, I think it was GWAS and some proteomics and maybe some gene expression marker data, each of which would give you, like hundreds of quote unquote answers, and sort of, the real answers in there buried with a bunch of false positives. But ultimately what happened in this paper is he showed that, sort of there was one overlapping gene in all three of these data sets, and he ran some assays and determined, indeed that was the sort of the key to unlock this mystery. And that certainly worked well, if all of your data sets are sort of keyed to the same thing. But that's not the reality of biomedical data sets. There's, genomics measures, there's chemistry measures, there's phenotypical measures, there's different patient measures. And unless you're conveniently measuring them all from the same, patient population, over time, which is very expensive and very, very time consuming to do, there's really no easy way to sort of key all these things together. And my thought was like, maybe there is a way. And so the technology that I created and ultimately has been expanded upon is taking this concept, the concept of, that the answer to a very complex disease doesn't necessarily live in any one measurement or any one biomedical data set. And if you have the ability to ultimately pull in lots of very diverse, and by diverse I mean statistically independent data sets across a wide range of biomedical measures, and integrate them as a single processing unit, you can ultimately uncover things that other people essentially haven't noticed before, right? And then use that, in our case, to do lots of things, but in our case specifically to develop new therapeutics. So in all of our disease areas, ultimately what this means is we are working on new mechanisms of action, right? These are new, if you will, new concepts or new understanding of biology in these disease areas. And therefore what it means, or what the impact is to your earlier statement, is we're going after biology that potentially has a disease modifying effect that others have not, approached before. And therefore, sort of the promise of the opportunity is to make a significant dent in these very, complex diseases. And so that's a, kind of a high level view of what we do, but ultimately it's all about, integration of these very different data sets, and then using that to ultimately, come up with a new experimental medicine that we would, explore and experiment with, and see what it can mean for patient impact.
Yeah, I think that's one of the most exciting parts of when I talk to everybody. It's like, assuming the system is designed well and the data going in is actually good, and it's like, wow, I didn't know that happened. I didn't know that pathway was involved, or with this little, I didn't realize that tweak could make this difference. And so that's what I see when I talk to different people that are working in this area of, I just didn't know, or that's not what none of the papers talked about this, or that's not what I learned in school. And so that's the most fascinating part of these systems, where you can identify things that faster, hopefully, and more accurately, hopefully, than you might normally do with a human being. No knock to human beings, where all of them are valuable, but it seems the systems move at a different pace and can handle a much broader level of data being input into them. And so that makes me, bring the question that Andrew, you and I have talked about it of, if you had to put a time frame around it or something, is this shortening the time to discovery? And I think you and I, the last time we talked, you said it's about 3 years where I can shave off on the front. And then at some point when I have to get to a mouse, I have to follow the normal trajectory of that mouse. But, and if that's changed and you're finding other areas, I'd love to hear it. But Peyton and Tim, where do you see the speed or the financial impact of what you're doing? You're doing it because it's moving it faster, or you're able to identify something that you haven't, but it's better than X or Y that's already being done in the marketplace.
For us, actually, this is, we do things faster. We do, I say, improve in a lot of metrics, but it's actually, for at least my company, about designing antibodies that couldn't otherwise exist. So for example, the standard monoclonal IgG, there are many tools out there, right, to sort of, I would say discover initial molecules and sort of to optimize them. But you start getting into these kind of next generation or kind of Frankenstein antibodies, nanobodies that are a tenth of the size, or kind of scFvs, which are these fragments that are, part of CAR T therapies or other formats. They become more complex and people have trouble engineering them. And you can kind of run your imagination and say, well, if I had the ability to engineer things, what other formats would I conceive of, would I consider? Tiny, I would say antibodies like cell-penetrating peptides that can get into cells and sort of have all sorts of characteristics, but they're difficult to engineer. And so we actually, instead of sort of doing the same thing faster, actually think more about how could we expand the universe of what could be a potential therapeutic protein, and how could that solve current patient needs in ways that existing therapeutics do not. And we do that by doing things faster, sort of and cheaper and sort of more smartly. But ultimately, that's what we really care about.
I think my answer is probably somewhat like Peyton's. I think if you look at diagnostics and biomarkers in particular, right, a lot of diagnostics are about, hey, we found that if you measure this one protein, that's useful for health. So it's a very, very slow process, and it's not optimized. You tend to study things that are obvious because they're easy to measure. Or like in our field, there's one protein called procalcitonin that's sort of the current closest biomarker for whether or not somebody has a bacterial infection. But PCT, as procalcitonin is abbreviated, procalcitonin was discovered, 30 years ago, and it was originally basically by accident that someone even measured it in someone with bacterial infections, and then it worked pretty well. And it's just sort of it's based on serendipity, and it can't be improved upon. It has however good procalcitonin was yesterday, that's how good it's going to be tomorrow, and how good it's going to be the day afterwards. I think the benefit of data science in diagnostics really began with cancer. When you had sort of the wonderfully successful tests like Oncotype, showing how you could measure signals across complex diseases by integrating things from multiple biomarkers, and a lot of those were designed. And there again, the problem was that they took a long time to develop, and of course they take a long time to actually run, right? Most of, if you've ever had one of those tests done, it's like a week send out. You send some tissue to a company, it gets processed, you get your answer 7 days later. So one of the things we're doing differently, it has to do with the way that we gather and integrate data sets to empower faster discovery. And then the other really, so it's sort of kind of like Andrew, and the other is basically the ability to build new answers that haven't yet existed, sort of more like Peyton. And ultimately the hope is to create a feedback loop where, better and better versions of the test can be slowly released. And so over time, it's not just that you're sort of stuck with hey, procalcitonin is as good as it is. It's like, you're on Incept version 5 in 2030, and it's, now X% more accurate. And I think that's a real benefit to patients.
So you guys have been doing this for a while. Do you see the promise of big data and AI playing out the way that you thought? Or is it different than you thought? Now that you like jumped into the pool and you've been swimming in it for a while, and is it fulfilling the dream you had? Is it more exciting than you thought? That's where I'd sort of ask the question.
Yeah, it's a funny question. I think coming from very different industries, looking at where I was 10 years ago, I think I was very naive in what it actually takes to bring a drug to market. And I think in the very early days of the company, my prior startups, one of them I was in and out in a year and it exited, right? And so there's no such thing in this industry, right, to do anything like that. And so, part of it was whatever biased by my prior experience, but I think part of it as well is it's sometimes I think also hard to see how far things have moved along, and I think even in Tim's description is he was sort of talking about, well, this was state-of-the-art science, in decades past. The work he's doing today was impossible back then, right? So,
right.
There's sort of these steady incremental improvements, and I think part of what really is happening in the industry is that as the things to solve essentially are becoming exponentially harder to, for example, for high throughput screening, which is maybe the old way of doing things to find a hit, or exponentially harder for, sort of diagnostic tests or sort of blood tests to sort of detect, these nuances. You sort of have to bring in these technologies and these capabilities that are exponentially better, at solving those things. And so I think what happens is you can therefore characterize it in a different way. Is the time faster? Well, like compared to the old way? Well, of course, because those old ways just don't have a chance of being able to do these things. Like, is it cheaper? Well, yeah, because those old ways, again, just don't have a chance, But I think part of it is what is the pace of innovation? And that's, I think, kind of where the rubber meets the road and sort of like what is actually possible and what's capable. And so today, we talk about having 18 concurrent disease programs, and we've got a very small team, and we haven't raised very much money. That would just be flat out impossible 10 years ago. But now, and we still like raise some eyebrows around that. But now, sort of like, yeah, like, okay, we recognize, software is doing a lot of what used to happen in the wet lab. So this, sort of fits within the expectation of what, a modern technology company would do in the space. So I think there's that other angle of kind of like where expectations are kind of, catching up with what's actually been produced, and therefore, at some point we become the old technology, right? 30 years from now, some next generation will be talking about, those slow, painful people that, tried this in the past kind of stuff. And so it's, I think each iteration of innovation has its moment in the sun, if you will. And this is definitely the time for, the work that we're collectively doing.
I think the promise is ahead of us. I think it's, we're in an amazing time where I think things are starting to gain traction. We're starting to get tools and infrastructure. But I think that, if I were to say my conception of what machine learning and data science and generally computational powers can do in biology and medicine, I think it's just starting. So I'm excited to see things like AlphaFold. I'm excited to see a lot of, these kind of tools and capabilities be unlocked. But I think, you're solving a complex problem, right? That protein that you're predicting is in a cell, and it's part of a tissue, and it's part of a human. And there's so many more layers, I think, to consider. And I think it's, we're making great progress, and I certainly believe in the potential. That's why I'm here. But I do like to sort of say I think we're at the very, very early days. And as Andrew said, I think it's going to be fun to see what happens in 30 years. So I'm still very excited, but I wouldn't say we're at the accomplishments that I would consider of sort of really demonstrating the cornerstones of machine learning in biology and medicine.
I have to agree with Peyton. I think the best is ahead of us. So one of the courses we had to take at Stanford BMI, and I don't know if you two remember this, was Mark Musin taught this course on ontologies, but part of it had to do with sort of like the history of applications of sort of clinical data systems. And the oldest one, and like I forget the details, but it was in like the 70s, and it was around sort of clinical decision support for therapeutic prescribing. And then he, obviously that system isn't around today, and failed for its own reasons, and sort of walks through all of the failures of systems since then. And maybe one of the most remarkable things is how little AI and machine learning is actually employed in like most clinical practice. And there are some areas where for sure, like an area has fallen, but for all the buzz around computer vision, the AI that radiologists use most is probably their dictation, right? I mean, it's, it isn't yet sort of commonplace machine assisted radiography reads. And so will that be coming? Absolutely. But the interesting challenges in each successive generation of like, oh, we got pretty close, but it turned out that X wasn't good enough or wasn't built in the right way to be integrated with workflow, or is coming soon, but still needs some regulatory work or whatever else. There's plenty left to do.
I think that's probably one thing we all experience, actually transitioning right from academia to industry, where what's exciting in academia, is not necessarily what's going to be reliable, right? When you really want to make a good drug. So what you might think about it, be like, oh man, that's a really cool model. Like I'd love to try that. That's great. Then you kind of go right into industry and you're like, okay, well, this is going to matter. Right? This is going to go to patients. This has to work multiple times. And I think it is a very different standard, right? So I actually think it's the right thing. It's just because you find something to be very, very cool and kind of, I would say cutting edge, you really want it to work, and you want it to work over and over again. And I think there's an unappreciated gap between when something is first proposed or conceived of or demonstrated, and then when you can really make it work at scale, over and over again in areas that matter. And so I think, we're basically in that transition, right? For, I would say, a lot of these techniques in biology and medicine. Let's get it to work in practice. Let's get it to work in practice reliably. And now we can start sort of really seeing where we can move the needle on really impactful problems. But it's funny because I do think that's it's an important divide between sort of where we all started together in academia and kind of the papers that really paved the way to making things work day to day at our respective companies.
Yeah, no, I would agree with that. I mean, look, most of our focus these days is not on discovery. It is actually in the development of the therapeutics. It is about, preparing for IND filings. It's all the regulatory work we need to do that. It's medicinal chemistry. It's a whole bunch of things that are outside of the discovery process. And as we proceed to the clinic, more and more of our overall effort as an organization has, less to do about the core innovation that, created all of these assets, and more about the heavy lifting you have to do to ultimately get that product to market. And I think, to kind of tie it back to my previous comments, I think there's been a new generation of capabilities that has been created. I think, to what these guys just said, it's going to be a while until we actually see those things in the clinic. And to Tim's point about, computer vision and radiology, like there's a lot of good science that's already there and has been shown, experimentally to do a better job than obviously the human looking at those images. But yeah, it's going to take a while until, that becomes the standard. I am, my daughter was born almost 5 years ago now, but I was shocked to observe, even back then, which was only 5 years ago, that medical records were being passed from clinic to clinic with a fax machine. It just blew my mind. Like, you got to be kidding me! A fax machine? Like, I don't think I've seen a fax machine, right, in all these years. And so, I think part of it is, there's if you want to take the place where innovation moved the slowest, it's certainly got to be, government, healthcare, education. I'm not sure which of those might be the slowest, but there is a time for these new technologies to permeate the industry, and that is going to take time. And I think that's when ultimately patients and the people that are on the receiving end of all this innovation, like that's when they're going to see that difference. And it is going to take many years for this stuff to kind of, make its way through the process and ultimately, into the hands of providers and ultimately to patients. And that big benefit is going to come in the years to come, right? It's obviously, not in front of patients in many cases today.
Yeah, well, maybe it's, my brain is wired towards risk or innovation, because I'm like, well, if you wait till it's done to get involved, you're way too late. Right, you're either going to be a dinosaur or you're going to be obsolete, and we've seen that in a lot of areas of tech compared to old standard industry. There was a great piece the other day about this engineer at Ford who'd been working on, the gas engine for 40 years and then wakes up one morning and is like, I need to take early retirement because software and electric EV is the way it's going to go. And now I'm just in this sort of maintenance mode of what I'm doing. And I think about healthcare and I'm like, any institution that isn't at least dabbling in, using image analytics for radiology or something and starting to employ this and get used to this, I think they're way behind, where they may want to be in the next 5 years, because technology doesn't follow just a slow curve on the way up, right? It has a way to go straight up at one point, moving into an exponential curve. And I think the same for you guys. I mean, those companies that are not involved or partnering or investing in entities like you guys, is if you wait till it's finished, it's already too late, because Andrew, your system will keep kicking out new molecules, and Peyton, you'll be making new antibodies, and it'll be a little too late to catch up, right? I mean, that's the way I think about it.
Yeah, I would temper that a little bit, and the reason I would say that is because the companies that have been successful in the past in creating diagnostics and therapeutics, products are on patent, they have long life cycles, and they generate lots and lots of cash. And so, big pharma, big diagnostics companies, they can kind of wait around and sort of see how things shake out with different younger companies, and simply buy or acquire, assuming that the companies are willing to be acquired, those assets. And so I think large pharma has been very successful in becoming acquisition and essentially manufacturing and marketing machines. So
yep.
I don't necessarily think that some of these larger and established players, that their livelihoods are threatened. I think they will continue to acquire the best of the best with their large cash reserves. I think some companies in this space will gather the momentum and break out, and I think in time we might see some changes over time as to what the big sort of players are in this space. But unlike other industries, certainly software, it's like MySpace disappears and Facebook reappears the next day, right? And that's because you can deploy new technology and move users over in the course of an afternoon. And like from a therapeutics perspective or a diagnostics perspective, that's just not the pace at which those things move. So there's lots of room for that. And maybe similar in the automotive industry, you kind of have to build a factory and build some cars. It takes some time, right? So maybe there's some parallels there, I think, in some cases, but I don't see like a wholesale change happening overnight, at least from where I stand.
Not overnight, but we definitely have to have dinner and like have a discussion on this topic, because I would love to bring some examples to the table about how I see things. Once you digitize something, the model itself doesn't have to stay the same way as it used to be. It is up for change. So I think those are the shifts that may change the dynamics of the market. But I'd love to have that discussion with a wonderful glass of wine. After having come from Napa this week, I can show up with a few nice bottles. So yeah, thank you so much for taking the time to, Andrew, thank you for bringing this group together. Peyton, Tim, it was wonderful to meet both of you. I hope that we stay in touch, and I'll keep watching the companies as they progress, and I wish you guys incredible success.
Thank you so much.
Yeah.
It's our pleasure.
Andrew, Peyton, good to see you as always.
Absolutely.
You too.